So hello everybody, thank you. So as I can understand, I'm the last one in this conference, and I'm completely switching from the subject. So okay, so stay with me. So I'm going to talk about predicting the evolution of syntenies. Okay, so syntenies are genomic regions that are characterized by a certain conservation in gene content and order. And in this talk, I'm going to review some of the algorithmic methods for inferring the evolution of syntenies, which account for both trees, and gene orders. And actually, this talk is derived from a review paper that has just been published right now, actually, in Algorithms, in a special issue of Algorithms shared by Aida Wengrawa and Hélène Touzé. Okay, so initially the term synteny was just used to design genes on the same chromosome with a genetic distance that could not be determined, that were as close to each other, and for which genetic distance could not be determined by the frequency of recombination inferred from gene mapping, okay? So it was just designing genes on the same chromosome. But today, the term is largely used by biologists in an evolutionary meaning to characterize homologous genomic regions deriving from a common ancestral region. Okay, so for example, there are many examples of such regions of conserved genes. For example, operon in bacteria contain adjacent genes that are transcribed together into a single mRNA sequence. But there are many other examples. For example, many gene families are organized in parallogons, such as, for example, the genes of the opioid system. And I give here a figure of paralogous regions in human and chicken containing the genes involved in the opioid system. Okay, and you see, so there are 4 paralogous regions in chicken, 4 in human, and they are distributed among different chromosomes. Okay, so we have the evolutionary definition of syntenies, regions deriving from a common ancestral region, but of course we don't have direct access to evolution, so we have to identify them based on some constraints. So from a combinatorial point of view, the notion of 2 regions being synthenic has been defined in several ways, ranging from a strict requirement in terms of conserved segments with identical gene content, order, and orientation. So this is the first example here, to 2 permutation with exactly the same genes in the same orientation and in the same order. Okay, to a more flexible definition in terms of conserved content, but not necessarily in the same order, or even definition accounting for genes that are different. For example, here this region here is a 1 gene team. Okay, so the purple, gray, yellow and red genes here are present in the 2 genomes here, with a distance between 2 genes, okay, with 2 genes of the set that are not distant. The distance between 2 is not larger than 1 gene. Okay, you see you have the black gene here, which is in this sequence here, but not here. But anyway, the distance between 2 genes is not larger than 1 different gene. Okay, so the gene team definition of syntenies allow for some genes that are different, some foreign genes. Okay, so and more generally, they can be defined syntenies can be defined according to a set of considered evolutionary events that have led from an ancestral region to the observed extant ones. Okay, so for example, if we account for these events here, inversions, duplication, loss, substitution, So we can see, we can derive, anyway, we can see that these two sequences have derived one from the other by this type of events. So the definition of syntenies depends also on the events that we consider to infer the evolution of these two segments from an ancestral one. Okay, so when we want to study the evolution of these blocks, of these conserved blocks of genes in the genomes, we have to take into account various levels of evolution. Okay, so first at the gene sequence level, and from the alignment of amino acid or nucleotide sequences, we can infer the evolution of each gene family, so each gene in the syntenies, through punctual mutation, through substitution and indel. So gene trees reflecting this evolution of gene families are inferred from such, so from this such alignment, we infer gene trees, okay, reflecting the evolution of each gene family through punctual mutations. Okay, second, the organization of genes on the chromosome is reorganized through rearrangements. And the genome rearrangement field, so initiated in the 90s with the pioneering work of David Sankoff, Pavel Pevsner, and others, aims at inferring the most parsimonious or most plausible rearrangement scenario explaining the diversity of genes organization in present-day species. Okay, so the second level, when we study the evolution of syntenies, is to consider the order, the evolution of the order of genes in these conserved regions. And this is the purpose of the genome rearrangement field. Sorry. And third, genes also the content of genes in the syntenies is also evolved through losses, through gain and loss of new genes. So genes are gained and lost through various evolutionary mechanisms. So such gene movements are usually inferred one gene family at a time with the model of reconciliation, which consists in embedding the given gene tree in the species tree. So for example, here, so if we have this gene tree here, I'm sorry, I have trouble with the images. They are hiding my slides anyway. So for example, here we have this gene tree. So the tips are genes, and each gene, the A here means that the gene belongs to the species A. So here is the species tree, and this is a gene tree. So we have the gene A in genome A. We have two genes of this family in genome B, etc. Okay, so as we have 2B here, we see that at least this family has evolved by at least one duplication in genome B. So to infer the evolution that by gain and loss that have led to this gene tree, so we use reconciliation. So reconciliation consists in embedding the gene tree in the species tree and explaining the difference between the two trees through duplications, losses, and also horizontal gene transfers. I will talk later about horizontal gene transfer. So here these are duplications. The circles here are speciation events, and the dotted lines are losses. So we see that we can infer this evolution of the gene tree. So we had one duplication here before the speciation, the first speciation of the genomes, and a second duplication, etc., and losses. Okay. In part, minimizing duplication and losses Okay, if we ignore horizontal gene transfer and if we just want to minimize the number of duplication and losses, this can be done by a method that is known as Lca mapping. So how we do that? We map each node of the gene tree to a node of the species tree. So each node is mapped to the least common ancestor of the tips of this subtree. For example, here, this node here is the root of this subtree here. So the tips are C, A, B. So we look at the least common ancestor of A, B, C here. It's gamma. So this node is mapped to gamma. And then a node is a duplication node if and only if it is mapped to the same a node of S as one of its children. So for example, here, this node is mapped to gamma, and this children is also mapped to gamma. So this is a duplication node. The same holds for this one, and the others are speciation nodes. Okay, so from this LCI mapping, we can infer the minimum number of duplication and losses. So we infer gain and loss events through such a reconciliation mechanism, a method. Okay, so we have considered all this. You see, gene order. The problem that the problem is that, okay, so syntenies evolve through different events, but actually the rearrangements events are considered in the rearrangement fields, while gain and loss events are inferred from the reconciliation fields, accounting for gene trees. Here I will present, I review some algorithmic methods for inferring ancestral syntenies that account for both types of events. So such unifying framework are not so frequent. There are few of them that integrate both the gene order and gene trees in the same methodology. So my goal here is to review some of these methods. So for example, this figure here is an example. It is a simplified representation of the evolutionary history of tRNA operons in a number of bacillus genomes, which has been published by my group in molecular biology and evolution. So here we tried, okay, so we was interested in inferring the evolution of the content in the tRNA content of 50 bacillus genomes through various events. And you see, so here we took into account both rearrangements and gene gain and loss events. So as you see, we have inversions here, transpositions, which are rearrangement events, but also duplications, losses, tandem duplications. For this method, we considered an alignment strategy, and the hypothesis, the requirement was not to have overlapping events. So actually, we didn't took into account the gene trees for each gene families in this example. So actually, I will not present the method we use it to get these evolutionary scenarios, but I will present few others that integrate both rearrangements of order constraint and gene order and gene trees. Okay, and so in the review paper, I have many more methods, but here I will reduce to 3. So the DECO algorithms, the super reconciliation approach, minimizing duplication episodes. So I will talk more about duplication and losses here. And finally, I will show how we can generalize this, how this method generalize to horizontal gene transfers. Okay, so as we have just seen, reconciliations considers each gene family independently. So a first approach towards integrating the dependency information between genes is to account for gene adjacency. So the first effort to integrate gene order is to account for the adjacencies of two genes. Okay, so actually this is the goal of the DECO set of algorithms. Well, actually there are a few algorithms using this methodology. So these are the algorithm by Séverine Bérard, Eric Tanier, so teams from Montpellier and Lyon in France. Okay, so DECO integrate gene tree and gene order information for the purpose of inferring the evolution of adjacencies through gain and breakage. So, okay, so the algorithm does not account for multiple duplication. Okay, so anyway, so okay, so the input of the algorithm is a pair of event labeled gene trees and a set of adjacencies between the trees. And the output is a forest of adjacency trees of minimum cost, which determined the adjacency gain and breakage. So the goal of the algorithm here is to minimize adjacency gains and breakage. And considering an appropriate clustering of adjacencies, the problem is shown to reduce to a set of problems with exactly two gene trees, where all adjacencies are between two trees. So that's why the input is just two trees. Okay. So this is the input. So the input, so for example, here we have two gene families, the red and the blue. And we have adjacencies, so two gene trees, and we have adjacencies between the genes of these two families. Okay, so the first step is to reconcile these two gene trees, so to infer duplications and speciation event from the Lca mapping. Okay. And then, so here is, if we put, so this is the species tree, A and B, and then if we put the red tree here and the blue tree here, but now we are interested by the evolution of the adjacency. So this is an inferred history of the adjacency, of the evolution of adjacency. So this history here gives this output of the algorithm. So the output of the algorithm, I'm sorry, I have trouble finding a way to see my slide with my windows here anyway. Okay, so the output of the algorithm is two adjacency tree. So as you see here, so this adjacency here is duplicated here, and then we have a speciation, and but here the adjacency is lost because one gene is lost. So this induces the loss of this adjacency here, which we see here. And in this side, this gene is duplicated, but it remains just one adjacency here. So one of the duplicated gene remains adjacent to E7. Okay, and then this adjacency here is inherited in genome A and in genome B. And you see that this adjacency is a new one actually. So it's created it's a new one created. So as E6 becomes adjacent to E2 here. So the result here, so the output is a forest of two trees, okay, which reflects an evolution with one gain and no loss of adjacency. Okay. So to So a polynomial time dynamic programming algorithm is developed based on a set of recurrences detailing all the cases of adjacency, breakage, and gain, depending on whether only one or the two genes of an adjacency are duplicated or lost, together or separately. So the algorithm infers a set of ancestral adjacency. Okay, so so what we end up is a set of ancestral adjacencies. Okay, but if we want to infer segments, so we have to put the adjacency one after the other, but there is no guarantees actually that all these adjacency end up in a linear order. While this can be seen as a drawback of the method, actually the authors use it as a property, as a benchmark for correcting gene trees. Okay, and following the initial model, several extensions of the initial algorithm have been considered, accounting, for example, for horizontal gene transfer, or also handling fragmented extant genome assemblies. So, okay, so Art Deco, one of these version of the algorithms, also account for fragmented fragments of genome assemblies. Okay, and we have also a global score accounting for the gene tree likelihood, reconciliation cost, and adjacency gain and breakage cost was also developed by the authors. Okay, so this is a first set of algorithms that account for both gene trees and adjacencies, which are the first way to account for gene orders in order to infer ancestral syntenies. So another possibility is the super reconciliation approach. So this approach has been developed by our group. So in the reconciliation approach, as we have just seen, each gene family is considered independently, so assuming an independent evolution of each gene family. Okay, although this can be plausible for gene families that are far apart in the genomes, this is too restrictive for genes organized in centening blocks. So for genes that are close to each other in the genome. So for example, assume we have a segment, okay, so an ancestral region of 3 genes that has evolved this way. So these 3 genes has been duplicated. Actually, just the red and green part here of the synteny has been duplicated here. Okay, and then after speciation, they are both inherited on the 2 genomes. And then this segment here is also duplicated, and then we have the loss of these segmental losses here, and which give rise, so at the end, to this organization here in A, so 2 synteny in A and 2 syntenies in B. Okay, so this is again the evolutionary history, the true evolutionary history. So now, if we take each gene family independently, and if we correctly infer the evolution of each gene family, so this would be the tree for the red gene family. Okay, which then we can reconcile this gene tree, and which makes this labeling of internal nodes. So a duplication here, a speciation here, a duplication here. So you see this is related to this one, and this is related to this one. And then, so we can place it again in the species tree. And then for the blue, the blue gene family, no, oh, actually, we have also a green gene family. So this is the gene tree here that we can place here. And this the gene tree for the blue gene family that we can place here. And if we take each gene family independently, we end up with this evolutionary history, which is different from the actual evolutionary history, as here we account for single duplication and losses, while we have here segmental duplication and losses. So we have been interested in generalizing the reconciliation approach to a synteny tree, rather to single trees. So we have generalized the reconciliation approach to segmental duplication and losses. So in our methodology here, the input now is a set of syntenies rather than, okay, so a set of syntenies and a synteny tree and a species tree. And the output is an evolutionary scenario from a single ancestral synteny through segmental duplication and losses. Okay, so yes, but and here we have to, okay, and for a gene tree, as labels of internal nodes. Reconciliation just labels internal node by the type of event, but here actually we will need the type of event, but also the synteny, the actual synteny, and the segment that has been affected by the event. Okay, so the first challenge. Yeah, so as I was saying, so the input, so we need now, we don't just need gene trees, but we need a synteny tree. So the first challenge is to know how to infer such synteny tree. So this can be done in many ways. We can actually align, this can be done by aligning the syntenies themselves. Okay, so by doing that, the specificity of each gene family is ignored, in addition of being unsuitable in case of gene rearrangements that would obscure the alignment of syntenies. So rather than aligning the syntenies, we can also infer a synteny tree from the individual gene trees. So if the gene trees are consistent, then we can infer a super tree, which would be our synteny tree. And if the gene trees are not consistent, then the problem would to make them consistent. For example, reconstruct a consensus tree, minimizing a given distance with the set of input trees, or minimally correct the input gene trees, making them consistent. Anyway, so we can use many different ways to infer first an initial synteny tree. So then, so let's say we have now our synteny trees. So the input is a set of gene families, a set of syntenies in the different genomes, the species tree. Here the species tree is not difficult, as we have just 2 species, and a synteny trees. And the goal is then to infer a most parsimonious history of segmental duplication and losses. So we handle the problem by a 2-step approach. First, we label the internal nodes of the synteny tree as duplications or speciations following the Lca mapping. And second, we infer an optimal scenario of losses in agreement with this event labeled tree. Okay, so notice that such such an evolutionary history is not always possible, because if we allow just for duplication and losses, and if the gene orders are not consistent, we would not be able to infer such evolutionary history. So we have to, if we don't account for rearrangements, so we have to the gene orders of the syntenies should be consistent in some way. Okay, so this two-step method is exact for computing the duplication loss method. The main problem here is the second step, which consists in extending the tree with losses and infer the corresponding synteny and segment being duplicated or lost. So actually, the main problem is this second problem here. It's to infer ancestral syntenies. So actually, it's a small parsimony problem of inferring ancestral syntenies and the segment being affected by the event. Okay, so the required dynamic programming algorithm is of exponential time complexity due to the exponential size of the set of syntenies that should be considered at each internal node. And now, okay, so the problem, of course, is the fact that rearrangement are not considered. So ignoring rearrangement is, of course, too restrictive, and asking for consistent gene order is not very realistic. So a variant would be to allow for rearrangements. Okay, but we know that almost all versions of the small phylogeny problem are NP hard. So one solution would be to account for rearrangement by still only minimizing duplication and losses. So we account for rearrangement, but we only minimize duplication and losses. So this can also be seen as ignoring a gene order, actually. So for example, if we do that, we can infer such this kind of history here. For the unordered version of the problem, we don't ask for consistency of the gene orders here, and we will infer, for example, here you see from this ancestral segment here. So this segment has been duplicated completely, and then you see I don't care about the orientation here, the order here. So the order is different. So that means that going from here to here, we will need some rearrangements. And then this segment here is duplicated here. Okay, so this segment is duplicated, and the whole segment we find it here, and then this segment here is lost. Okay, so this is a history minimizing segmental duplication and losses. Okay, so we account for rearrangement, but we don't count them in the score. Okay, so this was another method allowing to include a way of inferring the evolution of syntenies, taking into account both the order of genes and the gene trees. Actually, the super reconciliation approach is just a generalization of the reconciliation approach, accounting for segmental duplication, segmental events. Another way of doing is to minimize duplication episodes. Okay, is to account for multiple genes. Okay, so another strategy is to account for multiple gene duplications, is to infer duplication scenarios, minimizing duplication episode. Actually, minimizing location in the species tree where a series of duplication may have occurred. Okay, so as input, we would have a collection of gene trees or event label gene trees and a species tree, and the output is an evolutionary scenario for the collection of the gene trees, yielding the minimum number of duplication episodes. Actually, this strategy has been used for the purpose of inferring whole genome duplication events, but it can as well be used for inferring a multiple duplication scenario for the gene families of a set of syntenies. It involves, two problems are related to minimizing duplication episodes. So according to an interval model, we have to assign to each duplication node D a gene tree, and I'm sorry, I still have a problem with this. So according to an, so we have two problems, okay? So the interval model and the clustering model. So according to an interval model, we have to assign to each duplication node of the gene tree an interval corresponding to the position in the species tree where the duplication may have occurred. And then according to a clustering model, we have to cluster the duplication into a set of minimum episodes. Okay, so as for the interval model, so it ranges for the most constrained one, restricted to the Lca mapping of D in S. So for example, you have two gene trees here and a species tree here, and the Lca mapping is just assigning to each gene here, to each duplicated node, its Lca mapping. So here it's A, here it's C, the least common ancestor here is F, etc. So this would be the most restricted, so the most constrained one, and the most relaxed one would be the FHS model, the Fellow, Hallett, and Stage model, where the duplication can be mapped to any node between the Lca mapping and the root of the species tree. So here, for example, this gene here, this duplication here may have occurred in the this branch A, or let's say here C. Okay, so the duplication could have occurred here, but also here and also here, etc. Okay, and between the two, we have the GMS model in green here, and the PG model in purple. So the PG model is the most relaxed one that does not convert the speciation into a duplication. So let's see, let's say, for example, take this node here, for example. So this node can be mapped. So the least common ancestor here is E. So the duplication could have occurred here or at F, but not at G, because if we assign G here, so then this node here would become, this speciation node here would become a duplication node. So we take the larger interval that would not change a speciation node into a duplication node. Okay, so this is the interval model. As for the clustering model, given an interval assignment of each duplication node of the set of gene trees, 3 different duplication clustering models have been proposed. So the episode clustering models allow clustering any 2 duplication. Okay, so this one so any 2 duplication that can be mapped to the same location can be clustered together. The minimum episode model excludes cases of duplication and an ancestor of this node in the same cluster. And for this one, the gene duplication clustering is like episode clustering, except that only duplication from different gene trees can be clustered. So if we take this example here, for example, let's consider the green assignment here and the arrow here. So let's take the assignment E for this node here, the assignment E for this node here. So this gives us this embedding of the trees in the species tree. So now the episode clustering models leads to these two duplication nodes. So all duplication nodes that can be assigned to the same position in the species tree can be clustered together. So we would cluster. This would consider all these as one duplication and this as one duplication too. While the minimum episodes model leads to 3 duplication episodes. So this would not cluster. In this case, this duplication is not clustered with its father here. So it gives 3 episodes of duplication, this one, and this one. Okay, so I have to hurry up, I guess. So the clustering problem can be formulated as a set cover problem. Alternatively, it can be formulated as a 3-interval cover problem, leading to polynomial time algorithm for the episode clustering and minimum. Sorry, I have to. Okay, so when we represent the problem as a 3-interval cover problem, then we get polynomial time algorithm for the episode clustering and minimum episode models under every interval model. As for this very loose model here, as for the most relaxed interval model, FHS model, the episode clustering has a trivial outcome with one cluster, while the gene duplication clustering and minimum episode models have been shown to be NP-hard. Okay, and finally, I will say something about horizontal gene transfer, but because until now, I just talked about segmental duplication and losses. Okay, horizontal gene transfers consist in the movement of gene from one species to another. So horizontal gene transfer are largely involved in shaping bacterial gene contents. Okay, horizontal gene transfer has been included later in the reconciliation analysis of gene families in the purpose of inferring scenarios of duplication, losses, and transfers, and identifying xenologous gene copy in addition to orthologs and paralogs. In this context, the DTL distance is the minimum number of duplication, transfers, and losses explaining a gene tree given a species tree. So for a transfer to happen from a source genome to a target genome, both genomes should have coexisted at the same time. So this requires the DTL reconciliation to be acyclic. So you see we have a gene tree here, a species tree. So this is a scenario with just a duplication and speciations. And here we have 2 scenarios with horizontal gene transfers. So as you see, this scenario is cyclic, so is undoable. Okay, as you see here. So this is cyclic, is not acyclic. Okay, so this is a cyclic scenario with 2 horizontal gene transfers. And an alternative and acyclic DTL scenario here with also 2 horizontal gene transfer. But this scenario is acyclic, but it is still biologically unfeasible as it is not date respecting according to the considered species tree. Okay, because you see here we have a transfer between one species and an ancestral species, okay, a species on another, which is not present anymore at that time. Okay, so the problem of finding a most parsimonious time consistent or acyclic DTL scenario is NP-hard. However, the problems become polynomial if the acyclicity requirement is dropped. In that case, the main idea for computing an optimal DTL reconciliation is to consider all possible mappings of the nodes of the gene tree to the node of the species tree using a dynamic programming approach. When a temporal ordering of internal node is available for the species tree, the DTL scenario must respect this ordering. A DTL scenario respecting a data tree is called a date respecting DTL scenario. So for example, this scenario here is not date respecting. So, okay, and notice that even, okay, so you can see the time, okay. Notice that even a date-respecting DTL scenario is not necessarily time consistent, as scenario may be locally consistent, but globally inconsistent. Now, global consistency may be obtained by subdividing the species tree into slices and exploring slices one after the other, and this can be done in time O(m) n squared, where m is the size of the gene tree and n is the size of the species tree. Okay, and now, so recently we extended the reconciliation approach to a special case of horizontal gene transfer. So this is a work that I did actually with Aida Wangrawa, who's here. So we extended the reconciliation approach to a special case of horizontal gene transfer, namely endosymbiotic gene transfers, where genes are transferred solely between the mitochondrial and nuclear genome of the same species. Okay, so exchanges are between the mitochondria and the nuclear genome of the same species. So such transfers actually have been a driven force in the evolution of eukaryotes since the unique ancestral endosymbiotic event integrating an alpha proteobacterium into a host eukaryotic cell. And the DLE distance, so the DLE for duplication, loss, and EGT. So this distance with this restricted type of horizontal gene transfer is easier to compute than the DTL distance, as there is no need of exploring a set of source genomes, nor there is a risk of time and consistency. So the linear time algorithm we developed for an arbitrary cost of operations can be seen as an adaptation of the quadratic time DTL algorithm for data trees, which allows transfer between any coexisting species. So this work has been published, accepted actually for the ISMB 2000, the next ISMB. Okay, so what about extending the DTL distance to super reconciliation, for example? Okay, so we have seen, so super reconciliation is the extension of reconciliation to account for segmental duplication and losses. So how to integrate segmental horizontal gene transfer? So because we can assume that segments of gene also have been transferred from one species to another. Okay, what we can see is that the two-step, if you remember well, our method was running in two steps. Okay, first infer the event labeling of the synteny tree, and then infer it was then infer the ancestral syntenies and blocks that are lost, etc. But this two-step methodology is not exact anymore for the DTL distance. In fact, reconciliation minimizing duplication, transfer, and loss is not necessarily a reconciliation minimizing duplication and transfers. because considering a node as a transfer rather than a speciation may decrease the number of required losses. So anyway, we can't just separate losses from duplication and horizontal gene transfer. So we have to have a dynamic programming algorithm accounting for all the events at once. And we are presently investigating this problem of the generating super reconciliation to handle transfers. Okay, so let me conclude my presentation now. Okay, so I could have presented other methods, of course, but anyway, despite the large effort that has been dedicated to the development of methods for inferring the evolution of synteny blocks, a lot remains to be done towards a unifying model, allowing to consider the variety of evolutionary events shaping the genomes. For example, in most studies, segmental movements, segmental duplication, for example, are indirectly inferred from single gene movements. So, for example, the DECO collection of algorithms stand on minimizing single adjacencies against all breakage. Okay, and then a linear order is inferred, but the basis is still single adjacencies movements. Okay. And also algorithm for minimizing episodes of events only indirectly refer to multiple duplications. So our super reconciliation model is more or less the only one accounting for segmental duplications. I didn't present algorithm for tandemly arrayed genes, but there are also algorithms accounting for both gene trees and gene orders to infer tandemly arrayed genes, but these algorithms are restricted to tandem duplications. And actually, presently, our super reconciliation algorithm doesn't account for tandem duplication. So we have still to improve it. So to account for tandem duplication, but also we have to find a better way to use individual gene trees. So as you see, okay, the super reconciliation stand on the inference of a nice right synteny tree. Okay, and actually we forget the individual gene trees, but what would be interesting is to actually account for more directly for these gene trees. We have also to find a way to include tandem duplications. We have to find a better way to integrate rearrangements. Also, in this talk, of course, I restricted the discussion to minimizing operation. But attributing appropriate score to the various event is an important issue. And the more event we consider, the more challenging is the problem of assigning appropriate scores. Also, an overall statistical framework for evaluating evolutionary hypotheses would surely be better than a heuristic outputting a single solution with no measure of reliability. Okay. However, the larger the list of considered evolutionary events, the more difficult is the problem of an appropriate sampling for such a statistical study. So being able to generate the whole set of most parsimonious scenarios remain an interesting approach for a probabilistic evaluation of the solutions. Anyway, so I can talk about many further improvements. For example, being able to have appropriate simulation for testing the accuracy of the developed algorithms is a problem by itself. So some events remain largely unexplored, as for example the endosymbiotic transfers. So this remains to be studied. So to summarize, accounting at the same time for sequence divergence So gene tree, gene order, gene content, evolving through punctual mutation, rearrangement, and segmental gain and loss events remain a challenge to be able to have a unifying framework for studying the evolution of synteny. Thank you. Okay.
Thank you very much, Nadia. That a very interesting presentation on your work. So you managed to present a very complete overview of all the various algorithmic framework that exists for the problem of reconstructing the evolution of syntenies. So thank you very much.
It's a challenge to talk alone in your office, you know, without knowing who is listening. And if you are saying makes sense anyway, it's a challenge. It's better to have the people in front of us.
Right, and I'm very sorry that I didn't have the chance to introduce you for your keynote talk. So I was in the room, but under the wrong name, so no one knows I was there. So I would like to take at least a time to reintroduce you correctly. So I will make it shorter. So for everyone, so Dr. Nadia El-Mabrouk is a full professor at the Department of Computer Science of University de Montréal. She holds a PhD in theoretical computer science from Université Paris 7 that she obtained in 1996. Nadia is a member of several centers, so I will only cite the Centre de Recherche Mathématique, which is a major hub for mathematics and related science in Quebec and Ontario. She's also a member of the Robert Sidagren Center in bioinformatics and genomics at the University of Montreal. So Dr. El-Mabrouk research domain is computational biology, and in particular, as we have seen, comparative genomics. So she and her lab have made very important, so key contribution to the field of comparative genomics over the years. So they have been working on the development of mathematical and algorithmical methods to reconstruct the evolution of entire genomes, gene families, and reconstructing ancestral genomes. So we have seen just a little bit of the work that was presented here for the evolution of synteny, so merging all those topics together. So thank you very much for your talk again. And so we have 2 minutes left for the question. I see that we don't have question in the Q&A. So if you have question, please, you can ask them now. So I will just start with my question. So my first question was about the super reconciliation. So like you said, when we come to rearrangement, almost all the versions of the small phylogeny problem are actually NP-hard. This is known, but there are also solutions like fixed parameter tractable algorithm that were explored for this problem. Would it be a direction to for the super reconciliation approach when we integrate the rearrangement?
Yeah, we can think about many ways to do it. Yes, sure. Yes, I don't know exactly what you are thinking about exactly, but yes, we have to anyway. What we have now is not satisfactory enough, and we have to find a better way to include rearrangements. Yes, sure. We can do fine.
Would it be possible that any tractable version of the small phylogeny problem can be actually extended to a solution to the super reconciliation problem?
Maybe. I don't know exactly right now.
I don't know what is the time that we still have. So 1 minute. So maybe I will just make a last quick question. So about the minimizing the duplication episodes, so the second part you talked about. So do they account for the gene proximity within the genome?
No, not at all. From what I have seen, no, it doesn't take into account the gene proximity. So as I said, the goal is was more to infer the whole genome duplication events to find times when individuals duplication have occurred, and that could be related to a whole genome duplication event. But no, I don't think there is a proximity I didn't see that, but we can use it anyway for the yes, for genes that are that are close to each other. Yes, so I don't see why we can't use this method for inferring the evolution of syntenies through multiple duplications.